Immunological Society, they celebrated a Day of Immunology on 29th April. With the great success of that event, this day was celebrated worldwide since 2007. This year, the theme is Immunology Talks to Public Health, which gives us an exciting opportunity to highlight the deep and broad impact of immunological research towards our society. In recent past, we faced COVID-19 pandemic. During this period, we saw the monumental achievement of immunologists and vaccinologists, which definitely highlighted the importance of immunological research for the benefits of public health. Harmony in our immune functioning keeps us fit and fine. And failure of that, we faces many diseases, not only cancer, autoimmunity, and allergy, but other diseases as well. The metabolic disorders, cardiovascular diseases, even neurodegenerative disorders. In today's webinar, NIPER S.A.S. Nagar is honored to welcome our distinguished speakers. I'm sure that they will share their sweet experience to work with immunology. Now, I would like to request our director, Professor Dulal Panda, to deliver his inaugurating talk. Sir, please.
Have you joined? Who is the hospital? Good afternoon. This is really a pleasure to have this webinar in NIPER S.A.S. Nagar. I welcome you all. You know, this is so important for us, this Immunology Day, that we need to listen from the experts, and probably many students will join. They will get a lot of understanding from these different talks. Today, if you start thinking about immunology, first thing comes in mind is the vaccine. So, you know, particularly COVID-19, that taught us that immunology is so critical. Ebola or other, you know, even TB, one of the speakers is going to talk about the TB vaccine. So immunology has a lot to offer. Today you think of, you know, immune system and microbiome interaction, you think of autoimmune diseases, you think of, you know, many things that where immunology is playing a major role. So we must have some basic understanding. And today we are fortunate to have 4 eminent speakers. So I'm not going to take any time to take, you know, time out from their schedule. See, I see the first speaker is already there. So I will hand over, and I think the students will have, student and faculty who are attending this meeting will have a very good time. And again, I'm grateful to the speakers to agree to you know, give this talk. So welcome you all.
Yeah, thank you, sir, so much for your words of wisdom. Now we will initiate the webinar. We will have the first speaker, Professor Javed N. Agrawal, a senior professor at IIT Ropar. He has made several seminal contributions in the area of immunology, of infectious diseases, and vaccines. He has received many awards. To name a few, he has received Shanti Swarup Bhatnagar Award. He is fellow of INSA, Indian Academy of Sciences, and National Academy of Sciences. Today, he will be talking about paradigm shift in the vaccination strategies aimed at eradicating tuberculosis. With these few words, I welcome Professor Agrawala to deliver his talk.
Okay, thank you very much. And I'm grateful to Director of NIPER and the staff members, and to give me this opportunity to tell you some of the work which I have been doing concerning development of tuberculosis vaccine. So I'm going to present some of our latest work. some of the strategies which may be quite successful in eliminating vaccine, in eliminating tuberculosis. So based on this, can I have my first slide? Yeah, can you make it full screen? Yeah, thank you very much. Okay, so today I'm going to talk about the radical changes in vaccination strategies to eliminate mycobacterium tuberculosis from TB endemic regions. So while we were trying to understand host pathogen interaction, that is interaction between tuberculosis bacteria, which is Mycobacterium tuberculosis, and the host, so we could see that there are a lot of changes or a lot of difference in interaction between the people who are living in TB endemic region versus the people who are living in TB non-endemic regions, especially the Indian population, the way they interact with Mycobacterium tuberculosis versus the people in non-endemic regions, especially European people. Further, we could see some changes between the host interaction of people who are living in European countries versus people who are living in Northern European countries, where people are least exposed to Mycobacterium tuberculosis, especially people of Sweden, Norway, Finland. So when we take lymphocytes and just expose them to TB antigens, so the people living in, for example, UK or Germany, France, they still react against the antigen of Mycobacterium tuberculosis, whereas the population that is residing in Sweden, Norway, and Finland, they don't react at all. That is, the immune system has not seen tuberculosis bacteria. Whereas, if you see the case of people living in TB endemic regions like India, so the immune response is quite different versus European population versus Northern European population. So based on this, we thought, what are the changes? What are real changes in environment and in the population which is exposed to Mycobacterium tuberculosis? So today I'm going to talk about work which is majorly done at my previous Institute of Microbial Technology, where I spent 29 years. And 2018, I moved to IIT Ropar. So can I have a next slide, please? So I would like to first acknowledge my team who participated in this work. Today I'm going to talk about the work mainly, which was done by Gautaman and Deep, Pradeep, and Satyababu. So next, please. So first of all, I would also like to thank my collaborators. So in this study, the lipidated peptide was designed by Professor David Jackson from University of Melbourne, and Dr. Sherwan Mohali. He helped us in providing some of TLR2 knockout mice, and some experiments were done in his lab. Dr. Bhalla, he created nanoparticles, unique type of nanoparticles expressing on the surface TLR2 ligand, whereas Dr. Janmeja from GMCH Chandigarh, he provided us patient sample. And I'm also thankful to CSIR, DBT, DST, and Australia India Strategic Research Fund, and NHMRC Australia and MHRD for providing funding opportunities to us. Next, please. So we were trying to do, trying to understand host pathogen interaction. And during this interaction, what I told you that the immune response was quite distinct in people who were living in TB endemic region. And this is some of the publication which we have published. Next, please. So based on this, we try to understand that, previous slide, please. So then we try to understand the vaccine, and we try to make vaccine, and some of the studies we did, we published and took some patents on it. And recently, Last year, we published another good paper in JBC in trying to understand that how this epitope-based nanoparticles can deliver vaccine and how can we activate immune system where BCG has failed. Next, please. So what we observe that how to engineer a unique and a novel vaccine that is effective in TB endemic regions where BCG has failed. So let me tell you one important thing, that BCG has worked beautifully in TB non-endemic regions, but in TB endemic regions, BCG has miserably failed. Rather, there was extensive clinical trials which were done in Chingalpad in Madras. For 10 years, they followed BCG vaccinated population. And after 10 years, when they observed that 10,000 people, they were vaccinated with BCG and 10,000 people, they were not vaccinated with BCG. And when the result, after 10 years, when they examined the results were quite surprising, that people who were not vaccinated with BCG showed better protection than people who were vaccinated with BCG, indicating that BCG has somehow failed to deliver protection in Indian population. So if you see some of the, we started, looking into some of the publications where we observed that why is the reason, what would be the reason that BCG failed in TB endemic regions. And some of the main problem which we observed was that the BCG lack many antigens which may evoke important protective T cell responses, which are present against MTB but not in BCG. Whereas research studies have shown generation of Th1 response to BCG vaccination, but over time the response is dominated by Th2 profile. And after BCG vaccine, mice, they're prone to reactivation of disease due to the inability of these T cells to adequately express different type of co-stimulating molecules. And BCG does not elicit optimally CD8 T cells, which play a cardinal role in protection. Right, so the most important thing is that ultimately people realize that there are these environmental mycobacteria. We have many environmental mycobacteria in our environment. They interfere with the processing of BCG, and that's the reason BCG could not optimally activate protective immunity. And it is very well established fact that in Indian population or any TB endemic population, BCG, it protects from the manifestation of childhood TB, but it fails to protect the adult population. The reason is that BCG failed to induce long-lasting immunity. Next. So next, there was quite a good amount of publication that in case of antigen presentation, people have observed that the BCG antigens, the presentation is inhibited by environmental mycobacteria. So this is one of the important feature people observe, that since in our environment we have environmental mycobacteria, like these are the cousins, sister brothers of BCG or MTB. So they interfere in BCG antigen processing, and that may be considered as one of the reasons that BCG antigens are not optimally processed and presented to activate T cells. And in case of TB non-endemic area, where there is no environmental mycobacteria in the environment, so BCG can easily be processed. So that is one of the reasons. And BCG also, an environmental mycobacteria, they specifically block surface export of mature MSC class II molecules on the surface of MSC antigen presenting cells. And that is the reason that these antigens, they are not presented to T cells. Right, so the ultimate thing is that environmental mycobacteria, they impair in vivo antigen processing by dendritic cells. And the dendritic cells are the most important cells, which are the only cell that can activate naive T cells. So that is the reason that we don't get optimally activated our immune system. Next, please. So that means any vaccine that requires minimum antigen processing will work in TB endemic population, and which require extensive antigen presentation may not work. Next. So the peptides can overcome the problem of antigen processing, since they can directly bind to MSC molecules and require minimum antigen processing. So the use of peptide vaccine is experiencing renewed enthusiasm owing to the advances in delivery, the stability, and the design. So the question is, can peptide-based vaccine be successful choice in TB endemic regions? Next, please. So as I told you that peptide can directly bind on the surface of antigen presenting cells with MSC molecules. So what does it mean? It means that the peptides, they don't require any processing. They can directly bind, and once they are not required processing, they can directly activate T cells. So another advantage of choosing synthetic peptides is that entire antigen, it has three important components. One component is that there are immunodominant epitopes that induce protections against the pathogen. Second is that it has immunosuppressive epitopes as well. So if immunosuppressive epitopes are expressed dominantly, then this antigen is utilized by the bacteria to suppress immunity. Second important component is that many of these epitopes, they share homology with our body protein. So there are always chances that when we get infected, there's a prominence of getting activated autoreactive cells. So it can inflict us with autoreactivity, autoimmune diseases. So the advantage of peptide vaccine is that we can precisely select T cell and B cell epitopes. And the epitopes are accurately defined, and we can avoid autoreactive immunosuppressive portion in the antigen. And as I told you, that requires minimum processing. But the important thing is the snag association with peptide is that they fail to elicit immune response in genetically diverse populations. So as far as experiments are concerned in murine model, the peptides work beautifully because the mouse MSC molecule is very limited, or we use inbred mice and the required adjuvant. Okay, so next please. So to overcome this problem, what we did, we tried to select promiscuous peptides. So promiscuous peptides are those peptides which can bind to many HLA alleles. And such peptides can stimulate population of diverse genetic diversity. So in the initial phase of study, what we did, we identified immunodominant antigens of mycobacterium tuberculosis, such as 16, 19, 30, 38, 65, CPF, CFP10, EZ6. And what we did, since the sequence is available, we tried to synthesize the most immunodominant epitopes. And we synthesized 141 immunodominant epitopes. And next, please. So the criteria was that initially what we did, we took a Buffy cohort of 20 PPD positive healthy individuals, and they had no history of TB, and they started testing these promiscuous peptides. So these were people from TB endemic region, that is from India. And then what we did, we developed ELISA method, a fast screening method of binding these peptides to HLA class 1 and class 2 molecules. We had homozygous LBCL lines, which were expressing, HLA, homozygous HLA 1 or 2. And we did competition with peptide of fluoride, which was biotinylated, and it was showing promiscuous binding to HLA alleles. And we selected those HLA alleles which are highly prominent in Indian population. So any peptides out of these 141 peptides which showed high binding to HLA1, HLA2, plus showed promiscuous binding. Number 2, they induced T cell proliferation. Number 3 was the secretion of interferon gamma and IL-17. So on this basis, we selected. And the second thing was that high affinity binding. So interestingly, out of 141 peptides, what we observed that, and these are the HLA alleles which are highly dominant in Indian population. So we could find, it was quite interesting that when we shortlisted this peptide, you could get only 2 peptides out of 141, which made the criteria of protection in TB endemic regions. And these were peptide sequence 2140 and 9110. And unfortunately, both of these were from the 16-beta protein of Mycobacterium tuberculosis. Next, please. So we identified the immunodominant epitopes, and further, we could also identify CD8 T cell epitopes, same criteria, and these were from latent active form of the disease. Next, please. So this is how, since these peptides were showing good immune response on people who were PBD vaccinated, ultimately, we would like to make these peptide self-adjuvanting, that they should not require any administration of adjuvant, because adjuvant is a big problem for human vaccination. So many vaccines are available for human, but because of the known availability of right type of adjuvant, many of these vaccines, they fail to reach up to human benefit. So what we did, we took help from David Jackson, and these are some of the epitopes which we could identify. And initially what David Jackson did, he took our CD4 epitope and linked this CD4 epitope with lysine, and then serine, then he put the PALM2-CIS. PALM2-CIS is a ligand for TLR2. And he put initially CD8 epitope, and then he conjugated both CD4, CD8 epitopes, and then put some more CD4, CD8 epitopes, like 4 epitopes that he could put over here. But let me tell you an important thing, that David Jackson, he took about, he generated 13 of these constructs, and all 13 they failed. Failed in the sense they were activating dendritic cells, but they failed to activate T cells. So it was, very too much of frustration. But at the end, we could see some light when the 14th construct which is generated. We could see that T cells, CD4, CD8 T cells, they were also activated. They were proliferating. They're getting differentiated. They were secreting cytokines. They were functionally active. Next, please. Next. Okay, so now the important thing is why peptide vaccine may be better than BCG. So the reason is now we can overcome the problem of antigen processing due to an interference by environmental mycobacteria or mycobacterium tuberculosis latency, or even BCG, since no processing is required. So there was no fear of preformed immune response generated due to exposure to BCG, because once you have immunity and you're injecting full antigen or you're injecting bacteria, we have antibodies. So what's happening in TB endemic population? These antibodies, they bind to whole bacteria and it mops it out from the immune system. So it's not available for the cells of the immune system to optimally process and present it to T cells, so T cell can develop long-lasting immunity. But the peptides, usually they don't bind, they do not, T cell epitopes, they don't elicit antibody response. So we could not see any antibody response against this peptide. It's a 20-mer peptide. So now we have selective epitopes and we avoided autoreactive suppressive portion in this antigen. Next, please. So why we have chosen PALM2 is because it provides adjuvanting activity. It's a ligand for TLR2. Now the peptide has self-adjuvanting property, and TLR2 is copiously expressed on dendritic cells, and dendritic cells, they are the important cells for activating naive T cells, and they differentiate Th1 cells, they differentiate naive T cells into Th1 and Th17 cells, and Th17 cells play a crucial role in protections against tuberculosis. Plus, if there is an interaction between differentiating cells and an dendritic cell, so it does provide a long-term protective immunity. Okay, next, please. So why disease? I've already told that DCs are the only cell that can activate naive T cells. Next, please. So advantage of this vaccine is totally synthetic. No adjuvant is required. It can activate CD8 cells and it skews immune system towards Th1, Th17 cells and can activate naive T cells. Next, please. So now that this is totally a synthetic, right? And first thing what we wanted to see is whether it can activate dendritic cells, because important thing is that if you remember in this construct we have two portions. One portion is palm-2/cis and another is T cell epitopes. So palm-2/cis is going to ultimately TLR2 ligand going to hit DC. The first thing is we wanted to see whether it activates DC. So I'm going to use some terms over here like F91, which is free peptide, L91, which is lipidated peptide. Okay, next, please. So interesting part was that when we immunize animals with pre-peptide versus lipidated peptides, and we isolated their dendritic cells, and we took T cells. So T cells, which are peptide specific, these cells we labeled with CFAC, which is a green fluorescence dye. So the advantage is if you see under a fluorescence microscope, you can see these cells, fluorescing cells. And these are, F91 is a free peptide, L91 is a lipidated peptide. So if you see lipidated peptide, this brownish color, these are dendritic cells, and green are T cells. So you can see many conjugates around dendritic cells, indicating that the dendritic cells, these T cells, were interacting more with such dendritic cells which are obtained from animals immunized by lipidated peptides. Whereas if you see the middle one, F91, there were very less conjugates, 1 or 2, right? If you see untreated one, extreme left, these are simply dendritic cells plus T cells, and no antigen was added over here. We could not see any interaction between dendritic cells, and indicating that at least this palm-2/cis, which we conjugated, is working, right? So another thing, just below you see T cell proliferation. So lipidated peptide, it induced about 55% proliferation of T cells. In contrast, the unstimulated cell only 3%. And if you see towards right, blue and green, these are the conjugates. So maximum conjugates we could see up to 8 between dendritic cells and the lipidated peptide immunized animals, and we could see IL-12. Right, high IL-12 secretion, indicating that if there is IL-2 in the environment, and if a naive T cell is differentiating, is going to differentiate into Th1 cell, because IL-12 is a differentiating factor for naive T cells. So we could see interferon gamma secretion. So interferon gamma is also one of the key factors for protecting us from mycobacterial tuberculosis. Next, please. So we could see, enhanced expression of co-stimulatory molecules. These are flow cytometry data, and we could see CD80. They were highly expressed in animals immunized with L91, CD40, CD86. They were all highly expressed, right? So, plus we could also see these MSC molecules. They were highly expressed in animals immunized with L91. And we could not see there were downregulation of immature molecules. That is, this vaccine, it has a capability, it was potent enough to activate the turning of immature MSC molecules into mature MSC molecules. Next, please. So now the important is whether these lipidated peptides protect us against, protect against tuberculosis. So next, please. So what we did, we immunized these immunized mice with lipidated peptides, second boost after 21 days. Then we rested for 75 days to see that it generates bona memory T cells. And then later on, aerosol challenge with H37RV, which is a virulent strain of mycobacterial tuberculosis. And then we did bacterial burden, histopathology, T cell proliferation, cytokine, and T cell memory markers. Next, please. And we did, like T cell analysis was done by intercellular cytokine staining. On single cell, we looked into the expression of IL-17, TNF alpha, interferon gamma. On the surface, we looked at CD4, 44, CD62L, 25, 27. These are memory markers on the surface, and intracellular cytokines. Next, please. So importantly, if you see here, there was a big, high proliferation, a very robust proliferation by L91. It was 52%. And as compared to free peptide, it was 16%. So there was a huge difference in induction of proliferation by the lipidated peptide. Okay, so, and we could also see the generation of memory T cells, which we observed in the middle panel through CD44 and CD69 expression. And this was done through flow cytometry. We could see that memory markers were highly expressed on L91 immunized mice. These are 13% memory cells were generated as compared to F91, which are 6%, right? Indicating that it is also inducing the generation, not only activating T cells for proliferation, cytokine secreting, but also transforming these T cells, effector T cells, into memory T cells. Next, please. Okay, so what we did, we wanted to see whether these T cells, they're proliferating in lungs or in bronchoalveolar lavage, or they are simply proliferating in spleen. So these animals were vaccinated and then we fed them BRDU. So BRDU get incorporated into the cells which are proliferating. So proliferating cells, they take up BRDU dye. And then we looked into flow cytometry and examined the expression of CD44, CD62L, CCR5. So these CD44, CD62L, these are the marker from memory cells, and CCR7 is the chemokine marker responsive for migration of cells. So importantly, if you see here that spleen, lymph node, and mesenteric lymph nodes, all of these, they were a proliferation of T cells. Each line here indicates a proliferation of one division of a cell. But if you look into lung and ball, you see this homogeneous population. So indicating that the cells, they are primed in spleen, lymph node, and mesenteric lymph node. That is, they are primed in lymphoid organs. After that, they proliferate, and then they reach site of infection, lungs and ball. Next, please. So then we observe that the animals which are immunized, they are not showing, they are showing induction of interferon gamma secretion. So this bar diagram, this ELISA data. So high interferon gamma production, cytokine response for protection from mycobacterium tuberculosis, but the cytokines like IL-4, IL-5, IL-10, these are not responsive protection, rather they predispose people towards tuberculosis. So we could not see any secretion, very teeny tiny amount of secretion by L91. Importantly, now you see towards right, interferon gamma secreting T cells. We could see through ELISA, and you can also substantiated our results through flow cytometry as well. So L91, it induced 25% of cells to secrete interferon gamma as compared to 10% of cells which were non-lipidated. And we could see that these cells, if you go towards the lower right panel, this is also flow cytometry data, we could see some of the cells, they were secreting both TNF alpha and interferon gamma, that is, they are multifunctional cells. So L91 induced cells which are producing both interferon gamma and TNF alpha, 36%, whereas F91 induced 15%. So indicating that on lipidation, it has increased the immunogenicity of same peptide. And it is secreting both of these cytokines which are responsible, and TNF alpha is equally important for protection against mycobacterium tuberculosis. Next, please. So we also observed memory cell generation. So if you see here in the upper panel, CD44, CD62L, we use these markers and could see that whereas F91 is inducing 12% of memory cell generation, L91 induced 34% of T cell producing memory. We looked into another memory marker, which is CD127. If you look third lower panel, so free peptide is induced, has shown a shift of MF5 130 expression, whereas L91 induced 315-fold increase in the expression of 127 molecule. indicating higher generation of memory cells. Next, please. Okay, now the important thing is that this when the animals, we looked into these the presence of bacteria in the lungs. So if you see x-axis, y-axis, it shows log 10 CFU. So this is log 10. And we could see that it was showing better protection than even BCG. So BCG, the difference was statistically significant between BCG and L91. And of course, with F91, it was tremendous difference. And placebo, you see here, placebo versus L91, it brought down the clearance of MTB from the lungs of animals. Next, please. So we could see, we took human blood and we observed that even in vitro challenge of human blood, there was very high expression of memory markers. So CD45, RO, and RA, these are human memory markers, right? So if you just compare F91 versus L91, upper panel green, so it increased in human PBMCs as well from 4% to 13% increase in memory cells. And let me tell you one important thing. When we challenge these cells with 16 KDA antigen, this is a whole protein. Whereas this 9110 is a peptide from 16 KDA, right? So you can see that the entire protein, full protein, was inducing 3%, whereas the immunodominant epitope was increasing the generation of memory cell 13%. So this is very interesting that it shows better immune response, even the whole protein. And if you see these dot plots, these are each dot here represent single individual, and we took some of these individuals and then we stimulated with L91. We could see better generation of memory T cells in human blood. Next, please. Okay, so finally, this guinea pig is a gold standard of protection of TB. So if you have done experiments on tuberculosis and not shown protection in guinea pigs, so the data is not really very encouraging. So ultimately, we did these experiments. And let me tell you important thing: whatever we did, animal experiments, these were done by third party. These were done at Central Jalma Institute of Leprosy in Agra. So there was another person who was doing these experiments. Jalma people, they were doing. So these guinea pigs, same way they were immunized, second booster after 21 days, 75 days later, aerosol challenge with TB. And if you see here, when we sacrificed these animals, did CFU in the lungs, so it was showing better protection than BCG. So if you see bar diagram, see just purple versus blue. So L91 showed better protection than BCG and far better protection than a free peptide. And we also took a lipidated peptide of flu virus. So this is unrelated virus, just to rule out any possibility of artifact. That this is not because of PALM3 or something, that this flu peptide, it is conjugated with PALM3 as well, right? So there is this protection is quite specific, so it has been further substantiate the data of mouse. Next, please. So we have got US patent on this, and we could also publish this paper in infectious disease. 2 papers we published. And you can see same guinea pigs. These are the lungs which were taken. You just look towards the right panel and see that L91 lungs are clear, that almost 90% clear of bacteria, no granulomas. And if you see BCG lungs, still there were some granulomas. The lungs are not clean as seen in case of animals which are injected lipidated peptide. So this was really very interesting. And this is the lower panel is a non-specific peptide from flu virus. It was lipidated peptide, and the lung was having a lot of granulomas in it. So this very well establishes the fact that it induces protection. Next, please. Okay, this is how this peptide works. So this is, for example, macrophages, which is infected with Mycobacterium tuberculosis. So these purple are Mycobacterium tuberculosis, and this is expressing on the surface MSC class 1, class 2, or TLR2. So this is a model showing infection, that macrophage or disease they are infected. And now when this vaccine, it hits the infected cell, how does it work? Next, please. So this is how it binds. It binds from outside. So it does not require extensive processing. And when it binds, it is binding epitope 1, red one, it binds to MSC1, CTL epitope, and CD4 epitope binds to purple one, which is MSC2, and the peptide PAM2 cis, which is adjuvant, it binds to TLR2. So this is how it binds. And this was the reason to get this confirmation. David Jackson, he struggled a lot. He has to synthesize 13 different analogs. Okay, so you might have noticed that even some of the bacteria, they disappeared when it hit this infected cell. Next, please. So next, these CD4, CD8 T cells, they interact with this peptide, which is presented in context of MSC class 2. Next, please. So there's a proliferation of now T cells, they secrete cytokines, and these cytokines ultimately clear off the bacteria from inside the cell. Next, please. Okay, so conclusion is lipid peptide vaccine may be a future prophylactic strategy to eradicate TB from TB endemic regions. Next, please. So ultimately, we wanted to take this peptide now for clinical trials and large-scale immunization and large-scale production. And we are also thankful to NIPER because they did toxicology studies with this peptide. Dr. Tiku, he did. And for that, we, of course, paid money to them. And what we thought, but the problem we face that Jackson, he could produce, he said, when we asked Jackson to produce this peptide in grams, so he was incapable. He said that my lab is, the facilities they are to produce in milligram. So it may be difficult. So for clinical trials, you need this peptide to be synthesized in GLP conditions. But the biggest problem was how to synthesize in large scale. So we were, struggling to synthesize in larger scale the purifications. So it was very difficult. So ultimately, we thought, okay, we didn't have in mind that we're going to reach to such success with this vaccine that is going to reach clinical trials. So ICMR was ready to fund. There were many commercial partners ready to come and help us with clinical trials. But then there was a big problem in synthesis, and David Jackson, he struggled a lot. But what we thought, meanwhile, while David was synthesizing, we thought, why not we identify some alternative strategy? And we designed, nanoparticles. Next, please. Can you make full screen? Okay, so what we did to overcome this large-scale synthesis, we designed nanoparticles. Next, please. So these nanoparticles were designed, these are chitosan nanoparticle, and we entrapped all the epitopes over here, CD4, CD8 epitopes, and plus latency associated epitopes, chronic epitopes, and there were many epitopes we entrapped. And then on the surface, it was PAM3 cis. So this is how chitosan nanoparticles they were synthesized. And then we did some experiments. Next, please. So we were quite amazed to see that these nanoparticles, which are represented over here with the E91NP3. So E91 is a peptide, N91, E91, L91, 9110, and N is nanoparticle, and P3 is PAM3 cis. So interesting part was that we could see good amount of proliferation with these nanoparticles. The synthesis of nitric oxide. So we took here dendritic cells, and then we induced, we wanted to observe the secretion of nitric oxide because NO is an important factor in protection against tuberculosis. So we could also see through Western blot that these dendritic cells, they were secreting high amount of inos, which is a protein responsible for secretion of nitric oxide. So nitric oxide plus this protein also, expression of inos was pretty high. So this indicates that at least this vaccine is stimulating dendritic cells. Next, please. So we could also see that these dendritic cells, they secreted IL-6, IL-12, and TNF-alpha. So this was very interesting that these cytokines were also secreted, important cytokines, which helps in protection and differentiation of cells. Next, please. Okay, so here we observe that these nanoparticles, they were working through activation of NF-kappa signaling pathway, and some of the signaling pathway we did to understand the mechanistic interaction or mechanistic pathway of protection. So we did some of these P38 and STAT3 pathway was observed. But the important thing I would like to show you over here, if you go towards right, flow cytometric experiment is that these nanoparticles, they could activate, they could not activate cells which were TLR2 knockouts, indicating that it is working to TLR2 pathway. And if you see this histogram lower right, so these particles, they were very prominently, very avidly taken up by the DCs, which were derived from wild type mice. But there were very less amount of nanoparticles, which were uptaken by the TLR2 knockout dendritic cells. So these nanoparticles we hooked with FITC, and we could easily monitor through flow cytometry. So indicating that it is mainly uptake is mainly through TLR2 pathway. Next, please. So we could also see the high expression of MSC class 2 molecules, co-stimulating molecules like 8640. Next, please. So this is an important slide where we could co-localize these nanoparticles in with the lysosomes. So this red is a lamp staining, and green is nanoparticles, and this is done with time point experiments was done. 1R we took photographs through confocal microscopy, 2R, 4R, 6R. So with 6R we could see maximum uptake and of these nanoparticles for undergoing processing. Next, please. So finally, we did experiments to see whether it protects like lipidated peptides. Next, please. So here we injected in animals, and within 48 hours, within 2 hours, we could see, if you see towards left, this is a whole body imaging of small animals. So this is a mouse 2 hours, lower left panel. And after 2 hours, these particles start dispersing off, and within 40 hours, we could see that from the site of injection, they dispersed into the whole body. And then we observed that there was these particles, they reached into, within 48 hours, they reached into distal lymph nodes as well as proximal lymph nodes. So we could track down through labeling of these particles. And important thing is that these animals, they showed the generation of memory T cells. So we immunized with a placebo, just see towards the lower right. So this is placebo, and the lower one is immunized with these nanoparticles. So as compared to placebo immunization, the DCs they took 24% in placebo, uptake was 24, whereas the DCs which were exposed to these nanoparticles were 53%. So indicating that the immunization of these particles, it increases the uptake of to dendritic cells and ultimately processing and activating T cells. Next, please. So finally, we could see high proliferation of T cells as well. So towards, if you see towards upper left, so unstimulated cells, they were 2%, whereas when we stimulated with the nanoparticles from immunized mice, they were 15.7%. So there were high proliferation of T cells, and these T cells, they produce interferon gamma, they produce IL-12, IL-17, TNF-alpha. That is, it is inducing the generation of Th1 cells and Th17 cells which are important for protection against tuberculosis, IL-12, important cytokine which helps in differentiation of cells towards Th1 cells. And it also secreted IL-6, which helps in differentiation of cells towards IL-7, towards CS17 cells. Next, please. So we could also see in this case that it induced not only proliferation of CD4 T cells, but it could also induce proliferation of CD8 cells. So if you see upper panel right and left, CD4 T cell proliferating, and second row is toward the CD8 T cell proliferation. So we could see that there's a high increase in proliferation of both CD4, CD8 T cells. Next, please. So finally, we immunize animals with this vaccine. And again, 2 booster we have to give. And then later on, we aerosol challenge these animals. And 25 days later, we sacrifice. And this is y-axis is mycobacterium tuberculosis counts in the lungs. And these are towards the extreme right is a decrease in mycobacterial counts in the lungs of these animals. So there was a significant decrease in the mycobacterial counts in the lungs. So there was a great clearance, good amount of clearance of bacteria from the lungs, indicating that this is working as well like lipid peptide was working. Next, please. So the conclusion from this is that MTB epitopes entrapped in nanoparticle displaying TLR2 ligand on the surface may be a future vaccine for TB. So next, please. So here it is. This is how it probably is working. Next, please. So this is nanoparticle, and next. So this is again dendritic cell. Next. So this is how nanoparticle it goes here. Next. So these are taken up by dendritic cells. Next. Next. So these are now, they go inside, they're processed. Next, please. And now they are chopped off, the nanoparticles. And next, they come in contact with MSC molecules and they're presented to CD4, CD8 T cells. Next. And T cells, they expand. There's an increase in the expansion in the pool of peptide reactive T cells that produce cytokines, and this is how they clear bacteria, they activate dendritic cells, macrophages, and other T cells. Next, please. So this is next. This we published, previous, please. So this we published last year in Journal of Biological Chemistry, JBC. Next, please. So conclusion that these lipidated peptide vaccine may be a future prophylactic strategy to eliminate TB from TB endemic zones, and ICMR is now helping us to go for clinical trials. Next, please. So thank you very much, and I'm ready to take up the question answers. You have to unmute yourself. You're not audible.
Hello?
Yes, you're audible.
Am I audible now?
Yes, you're audible now.
Thank you, Professor Agrawala, for your excellent talk. And now I request Professor Dipankar Nandi to address the participant. Professor Dipankar Nandi is a senior professor of Indian Institute of Science, Bangalore. He has made several important contributions in the area of immunology, which includes studying of novel microbial genes during stress and infections, and understanding of post-encoded immune responses. Professor Nandi has worked with Professor James Allison, who won Nobel Prize in 2018 for his pioneer work on anti-CTLA-4 immune checkpoint inhibition therapy. Professor Nandi has numerous publications in international peer-reviewed journals with high impact. Today, he will be talking about some thoughts on pandemics and vaccines. Professor Nandi, please.
Yeah, thank you so much. Thank you to NIPER and all the organizers for this invitation. I hope you can all hear me. If my slides can be put up. Let me see if I can also get the laser control on it. Do you want me to try and share? Oh yeah, it's coming up. Yeah, that's it. Yeah. Try and make it full screen. I'll see if I can share, if I can. Okay, fine. All right. Okay. So what I will do today is to give some idea on pandemics and vaccines. I thought it was a little bit appropriate on this occasion. But okay, next slide. Yeah, so before I get to that, I will talk a little bit about the special day, which is the International Day of Immunology, which is what we are celebrating. So each year on 29th April, the International Day of Immunology is celebrated. And the idea over here is that to emphasize or to spread the importance of immunology to common folks, to lay folks. And I think as immunologists, we have a certain amount of duty to do this. And the 2023 Day of Immunology is, the theme is Immunology Talks to Public Health. And of course, there can be no better emphasis of this given the pandemic. So in fact, I have chosen, to talk a little bit about the pandemic and vaccines and give a little bit of a history and then talk a little bit about work that we did and the thoughts behind that work. Next slide. Yeah, so this is my lab collage, and we work on different aspects. We work on microbial stress responses. We work on T cell activation. We work on inflammatory models of disease and try and see what happens. But actually, I'm not going to talk about any of this in today's work, today's talk. It will be something completely new. So if I can have the next slide. Okay, so the motivation for the talk is as follows. We have recently had a once-in-a-century pandemic, so I'll address this event. And all the materials for this talk were actually prepared during the pandemic because of the circumstances. And I personally, got to know a lot of things, although I should have known it, I suppose, right? So I thought I would share all these aspects with you. I also felt it was important to do this now because the future is sort of somewhat uncertain. So we will see how it goes, and so I hope people will learn a little bit from this talk. Next slide. Okay, so this gives you a list of some recent pandemics. Okay, as you can see, we've had several in the recent past. So one of the first ones is the famous flu, the original H1N1 flu pandemic, which was 1918 to 1920, which is what a lot of people talk about, right? And in that during that pandemic, what was seen is that the young were more affected than the old. And so why is it that the young were affected more than the old? So the thought is the older populations must have been exposed to a sort of a similar flu virus, as a result of which they were protected, whereas the young were a more naive population and were more affected. The second one is the AIDS pandemic. Now, I have a somewhat of a personal connection with that, and that's because when I was doing my PhD, the AIDS pandemic was rampant, and in fact, we had a person in our laboratory, Peter, in fact, who unfortunately succumbed to the disease, which is really unfortunate. And when you hear about something, it's one thing. When a close person in the laboratory or in the family passes away because of a certain disease, it certainly hits you. And so I still remember, that event. And of course, lots of people in during that period passed away because of this. What often happened in the AIDS pandemic is a lot of the people on the community sort of migrated to the coast, either to the East Coast or West Coast, and we were on the West Coast and the Bay Area. Unfortunately, again, a lot of people, especially in the Bay Area and Los Angeles, were affected. In fact, Los Angeles was the first place where AIDS was discovered. Um, and then finally, there is the Ebola pandemic. Now, Ebola is interesting because I have been teaching immunology for several years, and among the last classes, one of the diseases that we would sort of pick upon was a viral hemorrhagic disease known as Ebola, and the idea was that that it may happen, right? So what will happen if this sort of happens? And it indeed happened. So in fact, during one lifetime, this indeed happened. So that was, unfortunate, but also important for our learning process. And then, of course, was the SARS pandemic, which, where we have, more than 6.8 million people dead. And that was, of course, something that most of us did not foresee. Next slide. But in all these pandemics, one of the major disasters is something that is somewhat overlooked and that is not discussed. And this occurred during the discovery of the New World. These paintings show is, the Europeans are being welcomed by the natives. And unfortunately, the natives are a naive population. They had not been exposed to many of the diseases like smallpox, typhoid, and so on. So unfortunately, when the Westerners sort of came in, the Europeans came in, they brought in themselves, they brought in their greed, but they also brought in microorganisms or pathogens that the natives were not exposed to. And unfortunately, a vast population were decimated. Next slide. Um, and in the next slide, it shows that, American Indian is sort of confronting some of the British generals with the fact that, they had given them a coat which was contaminated with smallpox, which is really unfortunate. So you can see somehow this part of history is not that well known and appreciated and does not get discussed. So which is an, very important aspect. Next slide. Okay, so I will now come to vaccinations, and vaccinations are a very important public health policy, and this, of course, can be seen by smallpox and also the polio ones. Now, polio has been eradicated in most except two countries right now, which is Pakistan and Afghanistan, and unfortunately, the problem over there is dissemination of the vaccination process, so which becomes important. The other small aspect that I'd like to talk about is on smallpox. Now, even before the smallpox vaccine came into being, people in the East, especially Turkey and India and so on, people knew that if you take from the pustules a small amount of this material and then give it to someone else, that that they could be protected. So in fact, they were known as tikka, and therefore the term tikka and tikkadas came up. So tikkadas were people who went around disseminating this. Now, one of the problems, of course, was that quality control was sort of missing, and so in case people got a higher dose, they came down with a severe form of the disease, and smallpox has about 30% mortality, and so on. And then, of course, you had Jenner, who discovered discovered the vaccination process. Now, what is interesting over here is that Jenner discovered, that people who suffered from cowpox did not come down with smallpox. So he realized that there was something over there, and the same idea about the pustules was used over there. And then the cowpox vaccination was becoming a problem, and so shifted to horsepox and so on. And these were all, in organisms where these viruses were grown at that time. And then somehow during the process, when we moved into cell culture and so on, ultimately there was the vaccine, and then you used, the vaccine for this. And but the virus changed. It was no longer vaccinia. It was in fact, yeah, it was no longer variola. It was, vaccinia. And so nobody knows where, this virus sort of came in. And this is what was used for a smallpox vaccination again. So something very interesting. So again, there were problems. Quality control over there and molecular diagnosis was not so well known. So this was an important aspect. Next one. Okay, one of the giants in the vaccine is Maurice Hillman. Again, very well known because he's the person who discovered more than 40 different types of vaccines, right? And there are some aspects about Hillman. So Hillman was not an academic. He worked, in fact, in a company, Merck, and then generated all these. And he was the head of the vaccine part over there, and was very successful in generating a lot of these vaccines. And I will tell you a few stories about Hillman, although most people are unaware of his contributions. Next, one of the one of the most famous stories about him was, his daughter had come down with the mumps virus, so he realized that she was suffering. So he went to the lab, got some instruments, isolated the virus, grew the virus, and then attenuated it. So the general idea is the daughter recovered from the infection, but not the virus. And so, so Hillman was obviously a gifted person, but he had his quirks. So he was a prickly kind of person. And in fact, he said that, I ran into conflict with just about everyone. So he's very picky, even though he would make the vaccines, the quality control part and all, he would go and check personally and all. And so that obviously led to interpersonal issues. One other important aspect, and especially for students, is that he was a strong proponent of the 7-day work culture for scientists. In fact, he said, "My hobby is working." So very important, no leave and so on. Let me now tell you a little bit about the person who made the first vaccine in India. And this was a person known as Waldemar Hafkin, who was trained in Pasteur Institute. And in fact, over there, he made the a cholera vaccine. Now, when he made the cholera vaccine, he had to sort of test it among, whether it was fine and all. So he injected himself, and he had fever, but he recovered and so on. But for human trials, he needed to go to a place where cholera was present, and this is where he went to Bengal. So from Paris, he sort of came and went to Bengal, and there he was looking for different groups, and so ultimately he found a cholera outbreak. So he immunized people. Next. Yeah, and he immunized people, and this is a famous photo of Hafkin inoculating people. So he had a control group, he had a vaccinated group, and the vaccine worked very well. So people got to know about him. He got well known for this skill. But there was another disease that was brewing. Next slide. And so this was, the plague that had come, and this occurred when again, this again started off in China, and then it came to Mumbai because you had the ships landing over here, and it spread. And this is carried by rats, and in fact, this led to the enactment of the Epidemics Disease Act of 1887. In fact, it was the same act which was put in use during the COVID pandemic. So it was quite a major act, and the idea over there was that the government could enter your house and burn it down because they thought it was infected with rats and so on. So it was quite a draconian one, but we need to be aware about this. So Hafkin was entrusted with trying to make a vaccine for this, and he worked in Grant Medical College, which is in Baikalla, Mumbai, and again he made the vaccine, and he injected himself again. And at that time, of course, we had no human ethics regulatory committees and so on, because doing all this would not be permitted these days. So he made the vaccine, and a lot of people benefited from it. he was knighted by the Queen. But Hafkin's stories are full of ups and downs. So this was a high for Hafkin, right? So he became very well known. In fact, the Governor General's palace was then used by him to make more vaccine, to scale it up and so on, so that it could be distributed, so that plague can be sort of controlled in India. Next slide. Right, and so this is what this. There's a previous slide to this. Yeah, so this is what happened. Unfortunately, there was a problem with one of the vials, and you had some people in Punjab dying because of post-vaccine. In fact, this was the first case of adverse events following immunization. So Hafkin was responsible for the first vaccine that was made in India. He was also, unfortunately, implicated in the first case of adverse events following immunization in India. So as a result of this, there was a committee that was appointed, and the committee implicated Hafkin because he was the director of the institute at that point. But again, there were other people who sort of supported him. Ronald Ross, for example. In fact, Ronald Ross, the Nobel laureate, he argued that Hafkin needs to be exonerated because otherwise public distrust in vaccines would grow. Yeah, next slide. So ultimately, vaccine was exonerated. The matter, went to the British Parliament, but he retired as head of the Calcutta Biology Labs, but he could not do any wet experiments, right? So he was not permitted to do any wet experiments. Only theory was allowed. And then he died he went back to France, and then he died over there. But given these enormous contributions of Hafkin, next slide, there was a movement to try and name the place where a lot of the vaccines were generated as the Hafkin Institute in Lower Parel, which still exists, in fact. And in fact, this institute it was also entrusted to try and make the COVID vaccines. Now, unfortunately, I'm from Bombay, and as someone who grew up in Bombay, I was completely unaware of Hafkin's contributions. I knew about the Hafkin Institute, but I had no idea about the history of Hafkin and the Hafkin Institute. And because of the pandemic, a lot of us, got to know about newer things, which is what I was trying to. Next slide. Okay, so I tried to make up for it. In April 2022, I went and visited the Hafkin. So I was not permitted inside because it was a holiday. But the guard over there did not want to take a photo. Did not want me to take photographs. But somehow we managed to take some photos over there. And just as a aside, I also visited another place in Bombay, which I did not know about, which is Gilbert Hill, which is made up of volcanic lava. It's about 200 feet tall, amazing place, and it's unique. And this is similar to Devil's Tower in Wyoming, but that is obviously a lot taller, which is about 800 feet and so on. So lots of things that we did not know sort of came up as a result of this. Next slide. Okay, so vaccinations in India are, by and large, well established by now, especially given the success of the smallpox vaccination. So there's a universal immunization program, and all kids are entitled to free vaccinations for at least 12 different diseases and so on. And the vaccination, and which has about 80% coverage. So. Of course, it would be, we need to aspire for so that all kids, all babies, get vaccinated and so they're protected. But 80%, given, the diverse country that we are and heterogeneous, is something commendable. And the photo over there, you can see that, it's a flood situation, but you see a small baby getting pricked. Baby doesn't like it, but it's, maybe good for the baby to be protected against different diseases and so on. Next slide. Okay, so I will now come to today's topic, which is COVID-19, which I will discuss some aspects that we studied. Next slide. Okay, it originated, in 2019, late 2019 in Wuhan, and that's how we sort of got to know about it. And we know that it's a respiratory infection, comes through contact, and you have both aerosols as well as droplets being responsible. And you have, most people get a mild to a moderate disease, but a few become critical and they need to be taken to hospitals and in some cases ICUs. And now, because of this, one of the important questions is why is it that some people get severe COVID? And one, there are several aspects to this, but one clear one which has arisen is people who have, for some reasons, inability to produce low amounts of type 1 interferons. Now, type 1 interferons are viral in nature, and if there are mutations or you have antibodies against type 1 interferons, it really compromises you. So that is one clear thing. There are other genes also involved, but type 1 interferons is one clear aspect. Next slide. And this was the famous paper from the Bat Lady in Wuhan Institute of Virology. And you can see that this paper in Nature was accepted in a record time, which is quite amazing. And it has been cited, more than 20,000 times, and which is again which is again amazing. And she is of course very well known for the discovery of newer viruses, and one of which was also this one. So I thought I would bring this out to students over here. Now, of course, COVID pandemic has been going on for several years now, and we have had a lot of deaths. And in India also, we have been affected by it. And just to bring some perspective into this, so officially, there are about 5.3 lakh deaths so far, and in India, for example, 1.5 lakh deaths occur each year to road accidents. There are 4.4 lakh deaths per year in India due to tuberculosis and so on. So just to bring that in perspective, and this is some of the original slides that we saw from New York, and which got us all scared in early 2020. And these are some black and white images, pictures of New York that were shown. You can see the streets are empty. People are inside because of the lockdown, and there's only one person around there going around with the PPE. So around 11th March 2020, WHO declares COVID-19 a pandemic. Around 16th March, the state government over here asked students to go back. 24th March, the government of India, announces lockdown. But it's only in June that the first COVID case in IST was recorded. So there was a certain lag. But once it came in, it again spread, and lots of people got infected. But that's the nature of this. Okay, so where do we come in? So we've been teaching immunology for several years, and one of the key questions that come up, and this is quite common, why is it that some people are more sensitive to allergies, infections, and so on? So this is a very important question in immunology. So everybody does not seem to have the same sensitivities. Some people are more sensitive, some people are less sensitive. So why is it? What is the difference? And one of the things I mentioned with COVID was, type 1 interferons, which play a very important role, and so on. Also, we keep track of, because of the teaching, we keep track of newer diseases. Now, because most of the students were asked to go back, there was only one student in the lab who was taking care of the cell lines and so on. And it was important to keep him academically active, otherwise you'd be constantly worrying. There were very few students over here, and they were, constantly being worried about, COVID coming in and so on. So just to distract him, we thought of, we need to do something else, right? And of course, no wet experiments were permitted. Okay. So there was one, there were some observations out there. So the virus originated from China, but as far as we know that, the reported deaths were, very less, right? But some countries, especially US, Italy, Spain, France, they had a higher number of COVID deaths, right? As opposed to, other countries. And also, in Bombay, for example, since, I'm sort of familiar, one of the big worries was what will happen to Dharavi, which is a crowded slum. And it was thought that would be a real problem. And of course, physical distancing in those places are almost impossible. But the impact over there was also a lot less than expected, certainly a lot lesser than the Western nations. So there had to be some reasons as to this difference. So we were wondering whether there are some aspects to this. And of course, there is the multiple factors involved in this, as this slide shows. There are different aspects. So one of the things is people with comorbidities, maybe also people that have an older population or an elderly population, right? They may be more sensitive to it, right? Whereas people with younger populations, they may be less sensitive. There are environmental factors. So maybe the Western countries that are sensitive to it are more on the colder side, whereas the warmer countries may not. And so you had different vaccination policies over here. You could have different cultural aspects. So in some cultures, for example, it's important to keep your shoes outside, wash your hands, and so on. Where in other cultures, maybe all this is not given too much importance. So there are a wide variety of possible reasons to it, right? So we wanted to see if we could address some of this. Next slide. Okay, so this was an observation that sort of we stumbled across. If you see, you have TB incidences as well as flu incidences. And the countries where there seem to be a lot of COVID deaths seem to have lesser TB incidences, especially Europe and Northern America. And also flu incidences, were a lot less over there. So, this was something that was there. I thought it was sort of more intriguing, and we sort of decided to follow up on this observation. Next slide. Okay, so, what are the effects? So basically, we framed the question that way. What are the effects of different respiratory pathogens on COVID-19 incidences and deaths? And so along with, pathogen exposure, do different vaccination policies, affect? So I was a little somewhat hesitant to get into this because it's a new area. But let me also say that, there were a lot of, controversies regarding this also. For example, there are a lot of controversies regarding BCG. So there are some people, who feel that BCG does not work at all. And I think Javed alluded to this, especially in endemic countries. For example, over here, the efficacy in adults has not been showed. He mentioned about the Chingle-Pit trials and so on. So there are people who are somewhat skeptical about it. But in countries where tuberculosis is not rampant, BCG does show effects. Now, importantly, BCG is among one of the first vaccines that are given to kids over here. And why is it given? Because BCG is a good pediatric vaccine. It protects the kids from tuberculosis, and that's why it's given. So again, lots of controversy. Now with flu vaccination, it's a huge enterprise. Especially a lot of the elderly get flu vaccination. So I knew this would be somewhat controversial and so on. But because of the pandemic, it sort of pushed us, to do something new, try something new. And the student was also game, so which is the best part, because this means, doing a lot of work on the computer, digging out data, and then trying to do it. So this was the general approach. What we did is we at that time, we looked at countries with, with a certain level of COVID deaths, looked at their flu incidences, looked at their flu deaths, looked at their TB incidences, TB deaths, and vaccinations, and did correlation plots to try and see if there are any correlations over there, and try and see if there is some sort of pattern to it. Next slide. So the other important aspect is we did it in a kinetic manner. Now, those of people who are sort of familiar, next slide, yeah, who are familiar with biochemistry would appreciate that it's better to follow a reaction in a kinetic manner rather than a single time point and a single substrate concentration and so on. So we sort of use the same policy over here. So you followed it in a kinetic manner, and as you can see on this is 2020 data, and you can see as with time, more numbers of countries are coming into the pool because COVID is spreading, right? And so what we found originally was, flu deaths negatively correlate. That means those countries that have a lot of flu deaths, there is less COVID deaths, right? So there's a negative correlation with this. Next slide. So why is it that some countries have less flu deaths, right? So, and that's because a lot of vaccines are given over there, and which is what I said. Flu vaccinations are a huge enterprise over here, over in some parts of the world. Next slide. And so we checked what happens with flu vaccinations. Those countries with very high amounts of flu vaccination, in fact, there's a positive correlation. So, more flu vaccination, less COVID deaths, and more, COVID-19 deaths. So that was the correlation. So why is it that this happens? So what is the role of flu infections over here? Next slide. So one is non-specific immunity. Flu vaccines, people have shown that if you give flu vaccines, you can also reduce COVID incidences, and all that is part of non-specific immunity that is done. So flu infections, and this is what others have shown. So this is correlation epidemiological data, but we are now trying to put in some sort of mechanisms into it. Flu infections are known to downregulate ACE2, and so maybe that's why it's tougher for the virus to get in. The third one that we invoked was viral interference, which is if one virus gets in, it interferes with the replication of another. Not true for all viruses, but some viruses, sort of interfere, right? Next slide. Okay, so then we looked at TB, and again, TB, what we found negatively correlates with COVID-19. Now TB, wherever there is high TB, BCG vaccination is given. Next slide. And so therefore again, more the BCG vaccination coverage, there is a lesser COVID-19 deaths and so on. All right. Now you can appreciate that there are lots of countries over here, and so it's a little bit of a mess. So what we did is we sort of segregated based on different aspects. Next slide. So let me just come back to implications of TB exposure again. Now how does this work? TB infections also are decrease the ACE2 receptor. Now BCG is known to induce strained immunity. So what is strained immunity? So this is epigenetic reprogramming. So one of the questions is, BCG is given when you're a kid, and how can it have effects much later on in life? So the idea is that it sort of epigenetically reprograms the immune response, and so you're able to generate cross-reactive immunity against pathogens, right? And in fact, this has been shown. BCG is effective, not only in tuberculosis, but it also lowers mortality in kids with pneumonia, sepsis, and so on. Next slide. Okay, so this is what I said is because there was a lot of countries, we segregated them. We segregated into TB high, flu low, and so on. And so what we found is those countries where you had high TB and high flu, right? Those had lesser COVID-19 deaths as opposed to where TB was low, flu was low, and so on. And let me also say the correlations that we found correlated well with COVID-19 deaths, but not with incidences. So that is another something that's important to keep this keep this aspect in mind. Next. Okay, and so again, countries with BCG and no flu vaccination have lower COVID-19, and this again we followed over time. So the thing is, BCG is protective, is somewhat protective, right? But it catches up, right? It catches up with time, and this is seen especially in the South American countries. In the South American countries, you have BCG, but there are less tuberculosis cases, but COVID-19 sort of caught up with them. Next slide. So then what we did is we segregated it based on BCG and TB deaths. And what we found is countries where you have high coverage of BCG as well as TB deaths, they are most protective. Then compared where you have BCG coverage but lower TB deaths. So that was really the important point. Now the role of BCG in COVID is also controversial. You have different people with different thoughts. But what we clearly showed is BCG is certainly protective, but it catches up, right? COVID catches up with it. But if you have BCG and now you have high COVID, then high TB, then you're better protected. So it's a somewhat of a dual thing that is required for better protection. So that's the important part over here. Next slide. Okay, so so the other important part is that this is a global study, right? So this is taking into all countries that had that threshold of COVID over there. And so what we found is people with high TB and flu. That means they are somewhat dirty because when we go to railway stations, when we go to airports, we are being exposed whether you like it or not. And for those of us from Bombay who have ridden the trains, the local trains, we have been exposed to quite a lot. But and so that may be helping protect us. Right. So, and BCG coverage also has negative correlation, and countries with BCG but no flu vaccinations have delayed transmission. But the important part over here is BCG coverage is important, but since it catches up, you need to be a little bit careful. Because and the countries with high TB or flu deaths along with BCG coverage turn out to be the most protected from COVID. Next slide. Okay, so anyway, I'm sort of, so basically what you are saying is, so BCG is useful, but perhaps what you need is continuous exposure to pathogens, and the two over there gives you higher cross protection, and that is, important in lowering COVID-19 deaths. So, that's what we saw in 2020. Next slide. So this has been published, and those of you who are interested can take a look into it. Let me now and emphasize some aspects. So our study looked at the role of respiratory diseases and vaccines, and we don't want to say that this we have sorted out everything. So this is part of the different factors that are at play, which is important over here. And so there are reports on flu vaccination and Mycobacterium indicus prani injections also, all of which are non-specific in protection, right? So that's an important. There have been clinical trials with BCG, and very interestingly, let me mention two of them. So the first one was from South Africa, and they did not show any that BCG had any protection with COVID. But you need to understand South Africa also has high amounts of tuberculosis. So again, BCG over there may not be showing. But BCG did show protection in the US, in America, where you have exposure to this is to tubercle bacilli is a lot less, and BCG may show an efficacy over there. So again, we are coming down to the fact whether where tuberculosis is endemic versus it's a non-endemic. So again, these are different aspects. But I also want to mention the controversial aspects about all these studies, right? So bottom line is not so clean environments may be useful, and so therefore in helping us protection, because as you get exposed, you're being protected against it. What we are doing now is we are looking at the 2021-22 data, right, and asking whether our initial observations that we made in 2020 still hold. And why is 2021, 2022 important? There are two important aspects with it. The first one is the rise of COVID variants, right? So with time, you have COVID variants coming up, and so we need to see whether the initial observations of ours still hold. The second is the role of vaccinations, because a significant amount of people have got vaccinated. Right? So we are in the process of analyzing the data, and in fact writing up the manuscript. So let me before I end, let me just show you not our data, but completely from an independent source, and that's the New York Times, and this is they have sort of normalized the data per capita. And if you see the cases per capita as well as deaths per capita, you can see that some parts of it were of the world were were a lot lesser, especially Asia and Africa seem to be a lot lesser, right? And you can let me also mention that Africa, for example, in terms of vaccinations, has done a poor job. So even though COVID vaccines have not reached, or the coverage has been poor, somehow Africa has escaped the onslaught of the majority of Africa has escaped the onslaught of COVID-19. So that's again somewhat of an interesting aspect, and this gives you a sort of a global perspective into what might be happening. Next slide. Okay, so let me just thank the people who did the work. So the student who was with me was Sanmoy Patak in my laboratory. So he was the only one there. So, it's not easy. He was the only student, and, it's a little bit lonely. Very few people on campus, and so I'm very happy that he sort of did the study on, and because we were entering an area which we are not familiar with. We got together with Mohit, who sort of helped us with the computational analysis just to make sure that we were doing. And there were lots of other people involved. I've listed them. And especially these people were important because they sort of egged me on to try and do something. Even though, we could not do any experiments, they egged us on to try and do something. And I'm glad that something came out. So after, something came out, I had to talk to different people also just to make sure that what we were doing is, not something totally off. So it was making some sort of a sense. So the top picture is of Sanmoy, and Sanmoy got married in December 2022, and that's him with his wife. Next slide. So this is snapshots of my lab, and that's in January 2020, where we were completely unaware of something that was coming by. And then it's in July 2021, and you can see we are all masked up. We have lost a lot of people because they've gone away, right? And then August 2022, we have removed our masks and we are sort of trying to recover. Next slide. And so this is IISc in April 2020. The gulmohars were in bloom. Gulmohars bloom over here at that time, and right now, and at that year, the gulmohar colors seemed to be, of a special nature. I thank you all for your attention.
Perspective about the COVID infection and also with respect to the previous pandemics and endemics which has affected the mankind. Thank you so much. And with the want of time, we would like to move on to the third talk. Third talk will be by Dr. S.A. Moli. And Dr. Sarachandra Moli is a renowned rheumatologist, and presently he is associated with Krishna Institute of Medical Sciences, Hyderabad. He has more than 35 years of rich experience in taking care and as a clinical immunologist and rheumatologist. He has conducted several clinical trials with leading pharmaceutical firms in the area of therapies for rheumatoid arthritis or SLE and other immune-related diseases. So now we will listen to his talk entitled Immunomodulatory Therapies for Autoimmune Diseases. So, sir, you can take over.
Sir, my screen is visible, sir.
Yes, sir, it is starting.
Is it visible now?
Right now, no, sir. Can again share it once more time. You can just close it once and again open and share it, sir.
Sir, is it visible now?
Not yet, sir. Now it is visible, sir. Yes. You can put it in.
Sir, I have some problem with my PPT.
Yes, sir.
I will do my best. So, thank you all, Dr. Stosh and the organizing committee for inviting me for this talk. So, I'm asked to talk about immunomodulatory therapies in autoimmune diseases. So if you see the inflammatory spectrum on the right-hand side of the slide, you can see the pure autoimmunity, the adaptive is big. That mediates the pure autoimmune disorders. And on the left-hand side, you can see pure autoimmune, where the innate immunity plays a role. Pure autoimmune disorders are like lupus and rheumatoid, and autoinflammatory disorders are like CAPS. And in the middle comes disease, ulcerative colitis, psoriatic arthritis, and ankylosing spondylitis. Now, quickly, we'll see what are the common autoimmune disorders we see in our day-to-day practice. This is rheumatoid arthritis. It's a very common autoimmune arthritis we see in our clinics. Rheumatoid arthritis is characterized by symmetrical polyarthritis. Symmetrical means involving both sides of the body. If the left wrist is involved, it affects the right also. So symmetrical inflammation, pain and swelling, redness of the inflamed joints on both sides of the body. To begin with, this functions of the hands and feet. On it affects the large joints. So this is called rheumatoid arthritis. This is the polyarthritis. The other one is rheumatoid arthritis or juvenile idiopathic arthritis in children 16 or below. 5 is childhood arthritis. The other one is lupus. 9 out of 10 patients in our clinic are female. So it's a female predominant disease. Full form is systemic lupus erythematosus. The short form also records as lupus. The main symptoms are, you can see a butterfly rash on the cheeks when they are exposed to sun, significant hair loss, recurrent mouth ulcers, and joint pains and swelling. These are the initial manifestations of lupus. Later on, it may affect other internal organs like kidneys, nervous system, central nervous system, or lungs, liver, and so on. So it's a multi-system disorder. The other one is psoriatic arthritis. You can see the psoriatic flakes behind the ear. And you can see here psoriatic arthritis affecting the finger joints. And also you can see the nails a bit ugly looking. Since psoriatic arthritis, a typical feature is not only affecting the skin and joints, it affects the nails also. You can see here nail pits. So this patient has got psoriatic arthritis. This is a case of dermatomyositis where the patient will have erythematous rashes on the face, eyelids, and swelling of the eyelids, face. And they will have proximal muscle weakness. That means when we are asked to get up from sitting position, patient will have difficulty. So this is called proximal muscle weakness. And CPK, creatinine phosphokinase, will be in thousands. So this is a case of dermatomyositis. Next is this is a case of vasculitis. One type of vasculitis called GPA, granulomatous polyangiitis. It affects the blood vessels in the nose, sinuses, throat, lungs, and kidneys. Here you can see the saddle nose. And here you can see the palatal ulcer and vasculitic lesions on the leg and feet. This is a case of scleroderma in a female where you can see here thickening of the skin, tightening of the skin, sclerodactyly. Again, it's more common in females. Another disease called Sjogren's, where the patient presents with severe dryness of the eyes, severe dryness of the mouth, vaginal dryness, and also dryness of the skin. This is another type of autoimmune disease, more common in females. You can see here, very dry tongue. Also, very teeth and swelling. Most of the autoimmune diseases are common in this type of example out of 10. So inflammation is pain more in the, and also pain and stiffness in spine, neck, and also later on they may have pain and swelling of the different joints of the hands and feet. X-ray, you can see here, sacral joints are inflamed, and you can see the sacroiliitis on plain X-ray. Genetically, 90% of them are HLA-B27 gene positive. So we discussed about the common rheumatological disorders across in rheumatology clinic. Now comes the therapeutics. What we call disease-modifying anti-rheumatic drugs.
Sir, slides, now they are moving.
We can divide them into synthetic and biologic. Synthetic, again, we can divide them into conventional, example, methotrexate, sulfasalazine, and targeted DMARDs like NIPS, tofacitinib, and baricitinib. When it comes to biologic, we can divide them into anti-TNFs like infliximab, etanercept, anti-IL-6, anti-B-cell, and anti-T-cell therapies. Biologics, they act through extracellular mechanism, and JAK kinase inhibitors act through intracellular mechanism. The commonly used drug in our rheumatology practice is corticosteroids. In the management of rheumatic disorders, there are two primary indications for their use in rheumatological conditions: to suppress the inflammatory cascade and for the modification of immune response. Mechanism of action, corticosteroids will have both genomic and non-genomic pathway. Why we call disease-modifying drugs? There is no cure for autoimmune conditions, but by using disease-modifying anti-rheumatic drugs, we can alter the course of the disease and we can put the disease into remission. We will discuss now the individual disease-modifying drugs. Initially, we discuss the conventional synthetic disease-modifying drugs. Anti-malaria, hydroxychloroquine is most commonly used rheumatology drug. Usual dose is between 200 to 400 milligrams. Mechanism of action acts by increasing the lysosomal pH. And also it decreases the production of interleukin-1, interleukin-6, interferons, and the prostaglandins. Sulfasalazine, usual dose is between 1 to 3 grams, maximum it can go up to 4 grams a day. It acts by upregulating the adenosine. It is the most commonly used drug for rheumatoid arthritis in Europe, whereas in US and in India, methotrexate is most commonly used drug for rheumatoid arthritis. Methotrexate acts by inhibiting ICOT transformylase imidilate synthetase, and dihydrofolate reductase. Leflunomide is another most commonly used disease-modifying drug for rheumatoid arthritis. Usual doses between 10 to 20 milligrams a day. It acts by inhibiting dihydroorate dehydrogenase. Azathioprine, the most commonly used anti-rheumatic medication for lupus patients. Usual doses between 1 to 2.5 milligrams per kg per day. On average, we use 50 to 150, 100 to 150 milligrams per day, depending upon the weight of the patient. Mechanism of action, by decreasing the synthesis of purine nucleotides. Mycophenolate, it acts by inhibiting the inosine 5 monophosphate dehydrogenase. Of late, we have been increasingly using this mycophenolate for lupus, lupus nephritis, and pulmonary fibrosis patients. Cyclophosphamide. Mostly we use this drug in lupus nephritis and CNS lupus. It acts the main effect of cyclophosphamide is due to the metabolite phosphoramide mustard. The phosphoramide mustard forms DNA crosslinks between and within DNA strands at guanine M7 position. This eventually leads to cell death. Tacrolimus, this is recently most commonly used drug in drug-resistant lupus, especially we use it in lupus nephritis and also in vasculitis. It's a potent inhibitor to T cell activation. Usual dosage is 2 to 5 milligrams BID. Eprimilast, this works by inhibiting the phosphodiesterase 4. Mainly it is indicated in psoriatic arthritis and psoriasis and also in Behçet's disease. Usual dose is 30 milligrams BID. The common side effect is diarrhea. The patient cannot tolerate, we decrease the dose to 20 milligrams BD or 30 milligrams OD. The next is target synthetic type of disease-modifying anti-rheumatic drugs, most commonly JAK inhibitors. These are intracellular proteins that JAKs are intracellular proteins that associate with and transduce signals from number of cytokines and growth factor receptors. There are 4 types of JAKs: JAK 1, 2, 3, and tyrosine kinase. The commonly available drugs, JAK kinase inhibitors in our practice are tofacitinib, baricitinib. Upadacitinib is not available in India. Ruxolitinib is the one studied in alopecia areata. You can see with this JAK inhibitor, you can see by week 16, you can see quite significant improvement of hair growth in the case of alopecia areata. And also JAKs have got important significant role in controlling rheumatoid arthritis. You can see here the American College of Rheumatology criteria, ACR 20, what we call ACR 20, 50, 70 response, with or without methotrexate. In all 4 JAK-NIB inhibitors, you can see significant improvement in joint scores. Next is IBIC, intravenous immunoglobulin. The usual dose is between 1 to 2 grams per kg for a period of 3 to 5 days. It's indicated in autoimmune thrombocytopenia, Kawasaki disease, dermatomyositis, polymyositis, CIDP and APS, hemolytic anemias. It acts by neutralizing the antibodies, modulation of B-cell rheumatoid, apoptosis, and providing anti-inflammatory cytokines, and also preventing and also treating the complement-mediated damage. Plasma exchange is very useful when the disease is aggressive and when rheumatological disease like vasculitis or lupus nephritis very aggressive, then finally we have to depend on plasma exchange. It acts by removing the immune complexes and autoantibodies that contribute to the pathogenesis of some of the rheumatic diseases. It acts by removing the cytokines, removal of immune complexes, replacing them with plasma components, and also by removing the pathological antibodies. It's indicated in thrombotic thrombocytopenic purpura, catastrophic APS, diffuse valvular hemorrhage in lupus, neuropsychiatric lupus, neuromyelitis optica, and ANCA-associated vasculitis. Rarely we use high-dose immunoablative therapy with autologous hematopoietic stem cell therapy in cases of drug-resistant lupus and scleroderma with significant results. Next comes biological drugs. There are different types of biological drugs available in the market, which we can divide them into anti-TNFs and cytokine monoclonal antibodies to interleukin 6, interleukin 1, B-cell therapies, and also antibodies against T cells. You can see here, this cartoon shows that different anti-TNF molecules like infliximab, etanercept, adalimumab, and anti-IL-6 processimab, anti-IL-1 anakinra, anti-IL-17 secukinumab, anti-B-cell therapy rituximab. Nomenclature used for biologic disease-modifying drugs. CEPT indicates a receptor drug which prevents a ligand from binding to its receptor, example etanercept. XEMAB, chimeric monoclonal antibody, example infliximab. ZUMAB, humanized monoclonal antibody, for example processimab. UMAB, fully human monoclonal antibody, example adalimumab, golimumab. RA, receptor antagonist, example anakinra. Now we'll see what are the common biological disease-modifying drugs available in the market. Etanercept, which is most commonly used anti-TNF molecule available in market for use of rheumatoid arthritis. Usual dose is 50 milligrams subcutaneous once a week. Infliximab is one of the anti-TNFs available. Interestingly, this infliximab is produced by, actually innovated by one of the Indian origin rheumatologist, Dr. Ravindra Mayani, when he was working in Guy's Hospital in London. Most commonly used biologic disease modifying drug in rheumatoid arthritis. Usual dose is 5 milligrams per kg once in 6 weeks. It's an IV drug. Adalimumab is another anti-TNF molecule. It's a fully human monoclonal antibody against the TNF. The dose is 40 milligrams once in 2 weeks. Golimumab is also an anti-TNF monoclonal antibody. And the dose is 50 milligrams once a month. And this is a monoclonal antibody to interleukin-1. These are also monoclonal antibodies to interleukin-1, but these are not available in India. Benelizumab is a monoclonal antibody against interleukin-5 used in bronchial asthma. Tocilizumab is an anti-IL-6 monoclonal antibody used in rheumatoid arthritis and also in giant cell arthritis. Secukinumab is one of the latest treatments for psoriasis and psoriatic arthritis and spondylitis. Is an anti-IL-17 monoclonal antibody. Rituximab, it is an anti-CD20 monoclonal antibody available. So it is anti-B cell therapy, mainly used for lupus and vasculitis. Is very effective biologic drug. A single dose will last for up to maximum 6 months. So usual dose is 1 gram on day 0 and again 1 gram day 14 and repeat after 6 months. Belimumab is the first biologic drug licensed for lupus. It's an anti-Willis monoclonal antibody. Abatacept is an anti-T cell monoclonal antibody approved for RA, psoriatic arthritis, and JA. Eculizumab is an anti-C5 monoclonal antibody indicated for hemolytic uremic syndrome and paroxysmal hemoglobinuria and myasthenia gravis. Avapan is one of the latest monoclonal antibodies available for complement 5A receptor. It is licensed to use in vasculitis like granulomatous polyangiitis and macroscopic polyangiitis. So till now we have discussed about pure autoimmune disorders where the adaptive immunity plays a major role. So now quickly we'll discuss about pure autoinflammatory conditions where you don't see any autoantibody. Here you see more innate immune activation in pure autoimmune, pure autoinflammatory disorders. Mostly they are seen in children. The common ones, for example, familial Mediterranean fever, where the children will have short episodes of recurrent episodes of 1 to 3 days attacks of fever, pleural effusion, pericardial effusion, arthritis, and skin rash. Rarely they will develop renal amyloidosis. The drug of choice is anti-TNF. The drug of choice is here canakinumab, which is interleukin-1 monoclonal antibody. The other autoinflammatory syndrome most commonly seen is TRAPS, TNF receptor associated
So thank you so much, Dr. Saraj Chandra Mauligi. And really, it was very informative talk about like what we can see, like a lot of newer therapies and their clinical implications. Also, you touched upon a lot of mechanisms. So it was very informative, especially for the researchers who are actually aiming for translational research. So with the want of time, we would move on to the next talk, sir. So thank you so much. So next we have a talk by Professor Oliver Paula. She is associated with Children's Hospital of Philadelphia of the University of Pennsylvania, USA. And her research focuses on defining newer mechanisms that regulate immune cell activations and protective immune responses. And her ultimate goal is to use this information and research outcome to design therapies which can treat or which can manage the immune-mediated pathologies found in autoimmune and allergic diseases. Professor Oliver received several prestigious awards and is author of scientific articles published in various reputed journals like Journal of Experimental Medicine, Nature Communications, Journal of Immunology, and various others. So with this brief introduction, I request Professor Paula Oliver to take on and present her talk.
Thank you so much for that very nice introduction. Can you all hear me okay?
Yes, we can hear you, Paula.
And can you see the screen with the presentation?
Yes, we can see.
Great, great. So thank you all for attending this this group of seminars today for the International Day of Immunology. It's great, that all of you all are here thinking about this really important topic. We have a great deal to do to really continue to define how immunology, how our immune systems work, so that we can better target the immune system for therapies to help prevent immune-mediated diseases and really help the immune system to do its job, which is to get rid of pathogens that we're exposed to. And so you've heard today about vaccine strategies and autoimmune disease, and really the focus of my research and the focus of this discussion today is on really a very different type of pathogen, a much larger pathogen, which is the worm, the helminth. And we're really focused on how the immune system really helps to get rid of these helminth infections, and how this can really, when these immune responses are poorly controlled, how they can lead to devastating effects of allergic disease. So about half of the world's population is either infected with a helminth or suffering from allergic disease. And both of these immune effects are really carried out by the type 2 immune response. So we've really evolved to have these type 2 immune responses to help to clear worm infections. And as I mentioned, when these type 2 immune responses are poorly controlled, they drive tissue damage and really devastating pathology that's seen in a lot of different allergic diseases. In the upper left panel here, you can get a sense of where the majority of these worm infections are occurring. This is really just focusing on soil transmitted helminth infections. And in the lower left panel, you can get a sense of the effect of how these type 2 immune responses can drive allergic disease, and how prevalent these allergic diseases have become. And really their prevalence is increasing over time, not decreasing. So we have a lot to do to try to really better define these type 2 immune responses and understand how we can really use this information to target the immune system to make it function a little more properly. So we're going to be talking more about these type 2 immune responses and exactly what they do. But the first thing I want to point out is that these type 2 immune responses are really focused on barrier surfaces. So surfaces including the lung, the skin, and the GI tract. And so we see quite a few of the allergic disease occurring in these sites. So in the lung, probably the most well-known allergic disease is asthma, which which is highly, which is a very common allergic disease in many countries. We also have allergic diseases that occur in the skin, including dermatitis. You see rash and hives as allergic responses in this tissue. And then the GI tract. So this includes everything from the upper GI tract with food allergy to the lower GI tract with inflammatory bowel diseases. So these barrier surfaces are really places where the type 2 immune response is focused. And we rarely see type 2 immune responses in other tissues or organs, like you see in autoimmune disease. And one of the reasons for this is because these are the surfaces, these are the tissues where our immune system is really trying to help protect us against helminth infections. And while we typically think of helminth infections because in the GI tract, because we know that that's really where the adult helminth lives and where it feeds off of tissues. There's also an important role of the lung and skin in anti-helminth immunity. And that's because many different helminth infections come through the skin and through the lung before those worms reside in the gut. So as is shown in this illustration, you start out with the secretion of eggs and feces, which then hatch and become larval forms. These worm larvae then often penetrate the skin and then go through the vascular system into the lungs. Let me get my pointer going here. They then migrate through the lung, through the vascular system into the lungs, where they're coughed up, swallowed, and that's the way they really enter the GI tract. So that really helps us to understand that the reason why we have these very prominent type 2 immune responses in the skin, in the lung, and in the gut. And so these are really the tissues where these immune responses are helping to protect us against worm infection as well as worm reinfection after the worms have been cleared. So what are these type 2 immune responses? How do they get initiated, and kind of what are the cellular features of these? So as I mentioned, the helminth larvae are the ones that are penetrating the skin. As these helminth larvae, migrate through the lung and migrate through the bloodstream and into the lung, when they enter the lung, they really cause quite a bit of tissue damage. And it's the damage that these helminths are doing that's really triggering the immune system. And so the epithelial cells in the lung really get damaged during the transit of the larvae into the lung. And these epithelial cells send out alarmins. These alarmins are really cytokines, which bind to cytokine receptors on a cell known as the innate lymphoid cell. This is a type 2 innate lymphoid cell. So before I get into exactly what those innate lymphoid cells do, let me kind of explain a little bit more about what they are. So most of you have heard about T cells. These can come in different types. They're Th1, Th2, and Th17 T cells. And these T cells kind of start out as these naive cells that then receive cytokine signals in addition to the stimulation they see through their T cell receptors. And that allows them to differentiate into these kind of subclasses of T helper cells. These T helper cells then secrete specific types of cytokines, which are listed here, and that helps these T cells orchestrate the downstream innate immune effectors that are important for the types of immune responses that are occurring. Th1 cells are really known for helping in antiviral responses. Th17 cells are more known for antibacterial responses, whereas Th2 cells are known for making cytokines that bring in cells, innate immune cells that help to clear worm infections. But what's more recently been determined is that there are an innate form of these T cells. So by that I mean cells that in many ways resemble T cells, but do not express T cell receptors. These innate lymphoid cells also look very much like the different subclasses of T cells, with ILC1s making similar cytokines to Th1 cells, ILC3s making similar cytokines to Th17 cells, and ILC2s making similar cytokines to Th2 cells. What's interesting about these innate lymphoid cells is that, as I mentioned, they don't have T cell receptors, but they also function very, very early in immune responses. Unlike T cells, which circulate around the body and help to look for pathogen infections in different tissue sites, innate lymphoid cells seed different tissues very early in life, and they are there for the duration. So they really sit in the tissues. They don't migrate much between tissues. And there are specific types of these innate lymphoid cells in different tissues, with innate lymphoid type 2 cells really being mostly focused within the barrier surfaces that we mentioned. So these innate lymphoid cells, as their T cell counterparts, respond very much to cytokine signals, but because they don't have T cell receptors, these cytokine signals downstream of alarmins are really what trigger the activation of these innate lymphoid cells. As I mentioned, these occur very early following helminth infection. These ILC2 cells then help to orchestrate the type of cells that are coming in to help clear the worm infection. So in the case of the ILC2 cell, they'll make IL-13, which helps to increase the mucus secretion from goblet cells to allow for the weep and sweep response that is seen during helminth infection, trying to get the worm, the helminth infection cleared by increasing the mucus levels of mucus and kind of sweeping the worms out of the GI tract. Additionally, these ILC2s can help initiate the T cell response by activating dendritic cells and T cells to make them better at kind of doing their job, which is to activate antibody responses from B cells and kind of ramp up the different cytokines that are made by ILC2 cells to increase the numbers of these innate lymphoid cells that help clear worm infection. And the main cell types that we know that help to clear the worm infection are eosinophils, basophils, and mast cells that really work in many ways downstream of antibodies to help actually make the environment so toxic for the worms that the worms opt to leave. So based on the information that these innate lymphoid cells really respond to cytokine signals, we study this ubiquitin ligase known as COL5. So COL5 is really the central hub of the CRL5 ubiquitin ligase. This CRL5 ubiquitin ligase uses SOX box containing proteins as its substrate receptors. And by substrate receptor, I mean the way that it recruits clients to the CRL5 complex. Several SOX box proteins are known to inhibit cytokine signaling by aiding the degradation of signaling proteins downstream of these receptors. As you all may be well aware, ubiquitin ligases really are key members of the ubiquitin pathway, where E1, E2, and E2 enzymes help to process ubiquitin and make it ready for transfer to a client protein. And it's the E3 ubiquitin ligase that really brings the client protein into the reaction, allowing ubiquitin to be transferred to the client protein. These ubiquitin chains can take several forms. In the case of CRL5, it's typically to add K48 type chains to allow degradation of the substrates or client proteins. So
So based on this information, we hypothesized that COL5 might regulate ILC2 cells and be important in helping aid worm clearance. So the first thing that we did was we looked at some publicly available databases, which from researchers who had done single-cell RNA sequencing on innate lymphoid cells, specifically ILC2s. So in this particular experiment, what they had done is they had isolated ILC2s from different tissues to look at the similarities and differences between the ILC2s that sit at different tissue sites. So this is a Leiden plot up in the upper left, and you can see that these different ILC2s from different tissues can be easily identified and separated from each other. And then you can look at what genes are particularly abundantly expressed in the innate lymphoid type 2 cells from these different tissues. And one of the things that was striking to us is that COL5 was abundantly expressed in the lung innate lymphoid cells, more so than the innate lymphoid cells in the GI tract, fat, or skin. And that made us think that maybe COL5 played a particularly important role in these lung ILC2s. If you look at the cytokine receptors that these innate lymphoid cells express, these ILC2 cells express in these different tissues, you can see that the IL-33 receptor is quite prominent in the lung, whereas it's not expressed very heavily in the gut or skin. And additionally, if you look at if you look at this, if you look at the different substrate receptors that COL5 might use, you can also see some tissue specificity in their expression, while some are not expressed as well in the lung as they are in the skin and gut. There are others that are expressed more prominently in the lung than in other tissues. So we decided that the best way to try to understand how COL5 might work in ILC2 cells during helminth infection was to make a model system in which to study it. And the model system we chose was the mouse. And what we did was we floxed, we added LOX-P sites to either side of some key exons in COL5, and then used a particular Cree that would allow deletion of COL5 specifically in innate lymphoid cells. And so in this particular case, this is known as the RED5 Cree. So this has the Cree inserted downstream of the IL-5 gene. So that what's been inserted specifically is an internal ribosomal entry site with an red fluorescent protein attached to the Cree. So in this particular case, when IL-5 is expressed, the IRES allows separate transcription of this protein production of this red fluorescent protein attached to a Cree. This Cree allows recombination of the LOX-P sites such that there is a deletion of the important exons, and it causes an out-of-frame mutation for everything downstream of that. So needless to say, the innate lymphoid type 2 cells in this particular mouse lack expression of COL5. So the first thing we wanted to see is whether or not these mice, how these mice tolerated worm infection. We decided to use a known model in mice known as Nymphostrongylus brasiliensis. So I'll be referring to this as Nippo infection. So the Nymphostrongylus actually has a life cycle very similar to what I just mentioned that occurs in many of the worm infections in humans, which is that there are eggs that get excreted in the feces, these that then can be kind of injected into the skin in the particular case of infecting mice. These larvae then move through the vasculature into the lungs. That occurs around day 3. They then move up the esophagus, get coughed and swallowed, and then enter the gut, where they usually live up to say 10 days to 2 weeks before the mouse's immune system is able to clear them. So if we look if we infect mice with the larvae, and then we look to see whether or not they're able to clear the worm infections, you can appreciate that mice lacking Col5 in their innate lymphoid cells, in their ILC2 cells, have fewer worms in the lungs on day 3. And if you look on day 7, you have far fewer adult worms in the GI tract. Interestingly, even though you see fewer worms in the lung, you actually, even though you see fewer worms in the lung and increased worm clearance, you see an increase in red blood cells in the bronchoalveolar lavage, which is what's sitting really in the alveoli of the lung itself on day 7. So this is a little bit surprising, because what this means is that there's continued damage to the lung, even though the worms are cleared very nicely. So either the damage is persistent or it's being worsened over time because possibly the immune responses that are going on in the lung. So based on Col5, the fact that we found that Col5 expression was high in lung ILC2 cells, we really focused our further efforts on what was happening in the lung itself during these early time points after lung infection. So we really kind of focused our efforts in the lung, looking in more detail on day 3 and also on day 7 after many of the worms have been cleared. So what was interesting to us is at these very early time points following Helminth infection, we saw increased numbers of neutrophils in the lung, as well as increased numbers of mast cells. Now, as I mentioned previously, it's known that mast cells are important in helping to clear worm cells in the lung. And so it was interesting to us that we saw both neutrophils and mast cells increased in the lung in the Col5 deficient mice. Not surprisingly, we did not see differences at day 3 in Th2 cells in the lung, as you wouldn't have expected the T cells to have responses this early after infection. But if we look, and as I mentioned, we've known for really decades that mast cells are important in clearing Helminth infection. What has been more appreciated very recently is that neutrophils can play a role in this as well. While this is still very early in our understanding of this, it has been shown in 2 different worm models that neutrophils can work together with certain types of macrophages in a process called netosis. And this is where they really kind of send out their DNA in order to trap the larvae and kill the larvae at sites of larval infection, worm larval infection. And so while we don't really understand exactly what's happening here, it is known that not only do these neutrophils have these very devastating effects on the larvae, but there's also a pretty big increase in the kind of tissue damage that occurs at sites where neutrophils and macrophages are using netosis to kill pathogens. And consistent with that, if we look at the histology of the Col5, the control and the Col5 red 5 mice, we see quite a bit of increased damage in the lung in the mice that lack Col5 in their ILC2s. So we see increased numbers of goblet cells, but we see also quite a bit of tissue destruction in these tissues. And to enumerate that over multiple slides, we actually looked at the PAS grade. So this is the PAS staining here, which shows the goblet cells in this really bright fuchsia color. And if we look over multiple slides, we can see that there's a lot more PAS staining, which means that there is continued inflammation in the lung even after Helminth clearance. So we'll get back to that concept in a little bit. But first, let's focus a little bit more on what might be happening at these very early time points after infection. So we were seeing changes in neutrophils and changes in mast cells. We looked more at the ILCs that were in the lung, specifically the ILC2s. And what was surprising is that we could see quite an increase in the cytokines that the ILC2s were making at these later time points on day 7, but we saw very little in differences in ILC2s at earlier time points. So we didn't see an increase in numbers of ILC2 cells at the early time points, nor did we see differences in their cytokine production. Now, I'm showing 2 different types of ILC2 cells here based on 2 subsets that have been identified for ILC2s in the lung. These include ILC2s that express the IL-33 receptor. So these are IL-33 positive ILC2s, and there are also IL-25 positive ILC2s. And while these are more prominent in the GI tract, they are found in the lung, but usually in fairly low numbers. They are thought to increase after some sort of inflammatory insult in the lung, particularly after worm infection. And it is thought that these cells really help to clear worm infections. But interestingly, we saw very few differences among ILC2s or IL-25 positive ILC2s, either at day 3 or day 7. Either in their number or in their cytokine production. So we were interested in the fact that we saw an increase in the IL-33 positive ILC2s at later time points, but we were really interested in what it was that these ILC2s might be doing on day 3 that wasn't just simply read out as an increase in cytokine production. And we decided to explore this more deeply by doing this single-cell RNA-seq, where we would isolate ILCs from the lung at day 3 after worm infection and really look at exactly what these cells might be doing, what was happening in these cells, and how this might differ in mice that lacked COL5. So we enriched for ILCs after helminth infection. These are the ILC2s shown here in the circled area. And you can appreciate that if you looked at wild type mice versus COL5 deficient mice, we did see more of these ILC2s on day 3 in COL5 deficient mice. And these we could identify as ILC2s because they were high for proteins that we know ILC2s express, and they didn't include expression of proteins that are more commonly found in other ILC subsets or in other types of immune cells that might have been enriched in this gate. So we really focused on these 3 subsets here that look to have the hallmarks of ILC2s. And we then subcategorized those. So we made subclusters of those ILC2s and found something really quite interesting. The first thing we found was that there were a number of different ILC2s, ILC2 subsets that were increased, that were more likely, I guess I should say, to be knockout rather than wild type. So in other words, if we looked at the subcluster of those ILC2s and the extent to which they were wild type, coming from the wild type mouse or coming from the knockout mouse, we saw expansions of certain clusters of these, more so in the knockout more so in the absence of COL5 than in the presence of COL5. And so some of these subsets were increased, but the one that really stood out to us was this cluster 1, this subset, the subcluster of ILC2s that kind of fell into this cluster that's shown here. So this is in wild type mice, and this is the frequency of that of those cells in the knockout mice. So if we look more into what these different subclusters express, we can see that the subclusters that show, increases in COL5, namely subclusters 2, 3, 4, 6, and 7, are all very high in IL-33 expression, which is shown up here in this top line. So these are IL-33 positive ILC2s, and we can see that those are increased in the absence of COL5. But if we look at this cluster 1, right, it does not seem to be positive for IL-33. And in fact, if we look at IL-7 receptor beta, which is the IL-25 receptor, it doesn't seem to be particularly high for that as well. But it is the most IL-13 expressing, that it is highest in its IL-13 expression of all of the subclusters of ILC2. So that was very interesting to us. It looked like COL5, the absence of COL5 was leading to an expansion of this particular subcluster of ILC2s that was very high in its expression of IL-13. And what was particularly interesting about this subcluster is it differed from other ILC2 subclusters in that, as I mentioned, it was low for IL-33 receptor, which is also known by its gene name of IL-1 receptor like 1. It was low in KLRG1, which is known to be very high in ILC2 cells, but it was very strong in its production of IL-33. And interestingly, when we looked at the differentially expressed genes that set this subcluster apart from the others, one of the most prominent things that we found was IL-18 receptor. Now, IL-18 receptor is not known to be abundant on lung ILC2s, but it is known to be abundant in skin ILC2s. So as I mentioned, ILC2s differ in where they sit in tissues. They're seated very early in life and they stay there for life. But these ILC2s actually have different types of receptors on their surface when they sit in these different tissues, with the idea that they respond to different things within the tissues, different types of cytokines, different types of alarmins. So skin ILC2s are known to have this IL-18 receptor positive phenotype, whereas lung ILC2s are really thought to be IL-33 positive. And as I mentioned, the gut ILC2s are positive for the IL-25 receptor, which has components of the IL-17 receptor. So we're really interested in kind of understanding what this expansion of IL-18 receptor positive ILC2s means in the absence of COL5. But what we really kind of are working from now is that COL5 is limiting signaling pathways downstream of both IL-33 receptor as well as the IL-18 receptor. And in doing so, particularly in the case of the IL-18 receptor, COL5 really helps to maintain the tissue identity of ILC2 cells in the lung, moving them towards being, kind of having a signal downstream of IL-33, but not IL-18. So IL-33 ILC2s are known to produce IL-13 and allow the recruitment of eosinophils in goblet cells. So this really helps to promote the weep and sweep response to clear helminths. And we really don't have a very good understanding of what IL-18 receptor positive cells do in the lung, but we're wondering whether it has any connection to this increase in neutrophils that we're seeing in the COL5 deficient, in the mice that lack COL5 in their ILC2s, and what role, and whether neutrophils are really helping to play a role in the removal of helminth larvae that we're seeing in mice that lack COL5 in their ILC2s. So the question then becomes, why exactly do we have COL5 in lung ILC2s if it actually prevents clearance of worms? And what we're thinking about that is, we see, even though we're seeing a reduction in the larva count in the lung, we do see an increase in the red blood cells in the bowel and an increase in the pathology, which makes us think that what COL5 is really doing is it's particularly abundantly expressed in the lung ILC2s to help prevent these really damaging responses in the lung. Because if you remember, the larvae only have a transient part of their life cycle in the lung. They come into the lung and then they, within a few days, they leave, they're coughed up, they're swallowed, and they go into the gut. So having a big immune response in the lung against those larvae may actually be more deleterious than advantageous. And so the idea here is maybe COL5 is helping to dampen these immune responses, these anti-larval helminth larval immune responses in the lung to allow that to really occur in the gut, where you can handle a little bit more tissue damage and as you're trying to mediate this worm clearance. So with that, I'd like to thank my lab, particularly Ipsita Guha, who really has driven this project forward with our collaborators from the Dabrowski Herbert Lab, Juan Inclin, Rico. They've really together just done some really, really exciting stuff in these ILC2s, and we're hoping to be able to translate this into future therapeutics.
Thank you, Paula. Thank you very much for your wonderful talk. And now it's time to conclude. All of us are really delighted in valuable deliberations of our distinguished speakers. They have nicely focused various aspects of immunology in context of different diseases. With the existing burden of various diseases like tuberculosis, dengue, and others, we have recently faced COVID-19 and autoimmune diseases like rheumatoid arthritis, IBDs, atopic dermatitis. They are also increasing alarmingly. But our speakers, they rightly pointed out that immunology research can hold the promise to solve this problem. But in recent years, we see different various achievement of immunology research, especially in the management of cancer. But to expedite this process, we need more and more participations from young researchers, young scientists, even doctors, by understanding the management or applications of immunology in management of diseases. And now I request Dr. Ashutosh to propose the vote of thanks.
So, good evening, all. Today, I feel honored and privileged to have been allowed to extend a vote of thanks for this wonderful webinar program. On behalf of NIPER, I sincerely thank the esteemed invited speakers for taking time from their busy schedule and gracing the occasions with their warm presence. It's indeed a great honor to host prominent speakers who are the big achievers in their field and have great contribution in the area of immunology health services. I would like to thank our dynamic director, sir, Professor P. Dulal Panda, who have been our guiding principle, helping us, imparting the wisdoms and nurturing all of us. Thank you, sir. I would like to thank all the faculty members, staff, and support members who have helped in organizing this webinar. A special mention to the IT support team for making the event successful. Last but not least, I want to extend a vote of thanks to all our delegates and participants from different NIPERs, from NIPER Mohali and other NIPERs and other institutes who have attended this webinar. So with this, we have come to end of this webinar. Thank you so much. Jai Hind.